didn't do that right. Okay, so here we go. And I didn't, when I said welcome to the people that are watching on the recording, I wasn't recording. So if you are watching this on recording and not with us live, hey, how you doing? All right, now I'll stop being lame and away we go. Now, those of you that are actually here with me in the meeting, if at any time you have a question, just go ahead, and if you put your cursor down at the bottom of the screen in the little chat box, then you can send me a little quick chat note, or you can also unmute yourself and ask me the question. So you just unmute yourself in the top right hand corner, or you can hit the space bar and that will also mute you. It'll take away the little microphone thing and put your name like mine is right now. Otherwise mine would have a muted microphone in front of my name. Okay, so there we go. So we're just going to go ahead and get into, I will put, I put this in a PowerPoint. So I'll also put the PowerPoint up for you guys to review as well. Okay, and those are going to pop up from time to time. Okay, so let's go ahead and get started. And I'm going to get rid of us, so we don't have to watch ourselves either. Okay, so welcome to our Monday lab on microscopic muscle anatomy, and there we go, and muscle physiology. And so we're just gonna go down through the list, and there will be times when we're gonna take a break, and oh shoot, there's a paper that oh, there it is. Okay, when we're going to take a break and watch some crash course videos, and then finish off everything. So let's just get started. All right. Okay, so the very first thing, you have this diagram in your lab manual. Can't remember which page it's on right now, but somewhere in exercise 12 of your lab manual, so you guys can go back and I highly suggest that you look at it, because now as far as our quizzes are going to be concerned, they will be from the diagrams that I give you either in the lab or on the PowerPoints like this, which you all have access to, but you need to be familiar with these diagrams. Okay, so we're just going to go to the next slide. When we take a look at skeletal muscle under the microscope, remember that it's striated with multiple nuclei, so we can see those there. But the most important thing we need to remember right now is the fact that it has striations. And so it has alternating light and dark bands. So if we take a look at the dark band and the light band, you can see on your list it says I band and A band. The I band is the light band, so this light area that does have a dark line in the very center of it, and then we go dark and then light and dark and light and dark and light and dark, all the way down the entire muscle fiber. And the reason why we do this is because the structure of the components within the muscle cell, so the proteins that are within the muscle cell, the ones that are found in the light band are very thin and allow light to pass through them more easily than those that we find in the dark band. And so if light can't pass through something on a microscope slide, then it shows up as darker than the surroundings. So then we know that the stuff inside the light band, there's not as much stuff, and the stuff that is there is much, well, we can say thinner, because it is thinner material, thinner proteins than what we find in the A band. But we're going to learn all about that today. All right, so when I take a look at a single muscle fiber or a muscle cell, so remember that a muscle cell and a muscle fiber are the exact same thing. So this is the entire cell. Remember that they are cylindrical in shape and run the entire length of the muscle. And so here on the outside, we are going to have a sarcolemma, is now the new name for the cell membrane. So on your list where it says sarcolemma, this is the cell membrane of the muscle fiber or the muscle cell now. And so this is what is polarized when we were talking about muscle cells before and membrane potentials. So this is where the ion channels are going to be, the ligand or chemically gated sodium channels, and the non-gated sodium and potassium, and the gated sodium and potassium, and those other ones I'm missing, the sodium potassium pumps. They're all going to be found within this membrane. And so all of the depolarization and repolarization, hyperpolarization is all going to take place within the sarcolemma. All right, now if we take a look inside of a muscle cell, that it's made up of a whole bunch of bungee cords. Basically they're stretchy, and they have the ability to contract and relax, and those bungee cords within the muscle fiber are known as myofibrils, which are made up of protein filaments called myofilaments, which we'll get further into in a little bit. But the muscle cell is a bundle of myofibrils that we find within the cell. So the actual shortening or contraction and the force that's produced that we have within a muscle is going to be caused by the number of myofibrils that we have within the cell itself. Now, one other thing about myofibrils is that the more myofibrils a cell happens to have, the greater the strength that that cell can produce. Remember that muscle cells or muscle fibers, you are born with all you're ever going to have. And so as we weight lift, as we do resistance training to become stronger, and we bulk up and we look like, you know, Dwayne Johnson or some other big bulk, Terry Crews or some big bulky guy, they weren't born like that, obviously. So how did they get so big and bulky? Well, as they continued to work out and have resistance training, their body responded by making more myofibrils, not more muscle fibers, because you're born with all that you're ever going to have. But those cells can get much larger in diameter or in circumference with increasing myofibrils. So the more you work out, the more you build myofibrils inside of your cells. And then you get big and bulky and buff and hypertrophied. But what if you can't contract your muscles anymore? You're paralyzed, or you're not eating and because you're starving to death or something. What's going to happen is that these myofibrils are going to be lost. And even if you were working out a whole lot and then you stopped working out, your body will consume the extra myofibrils because it's too expensive energy-wise to maintain more myofibrils than you need. So if you're not always lifting a whole lot of weight, then when you stop for really only about 8 weeks, you'll lose myofibrils. And the funny thing is, what takes 24 weeks to build up, you lose in 8 weeks. So that's kind of a sad thing. You lose it really fast. It takes forever to build. Okay, so so there's the sarcolemma. There are myofibrils. All right, let's go on to the next diagram. Now, because the myofibrils are the things that are going to be doing the shortening when told to by a neuron, or your brain is telling you to contract a muscle, we have to be able to tell the myofibrils it's time to contract. So it's easy to tell the cell that it's time to contract because we just depolarize the sarcolemma and action potential travels down the sarcolemma. But how do we let these guys know that are on the inside that aren't touching the sarcolemma? Well, we have along the length of the sarcolemma a series of tubes, which are just tubes that come in from the sarcolemma. I have girls being silly outside. I don't know if you can hear that. And so these are called T-tubules right there, or transverse tubules. And so they're going to go down into the middle of the cell, and they're going to come in contact with some material that's found on the outside of a myofibril called the sarcoplasmic reticulum. Now, sarcoplasmic reticulum is just a fancy name for smooth ER. And if you remember back when we were doing functions of the cell, and there were 3 functions of the smooth ER, and one of them was the storage of calcium. So that is their job inside of a cell, surrounding or covering over the myofibrils, is to store calcium that will be used at a later time that you will learn about, but not right now. So when it's time for muscle contraction to occur, then the action potential will travel down the sarcolemma, and then it'll go into the T-tubules, which will then cause the sarcoplasmic reticulum to depolarize, and then voltage gated channels will open up, and then calcium channels that are linked to that will then let calcium come out into the cytoplasm. All right, so we've done the first 4. All right, so now what we want to do is look even closer at a myofibril, at its structure, and see how it's made up. So we have repeating patterns down the length of the myofibril of light and dark bands. Okay, so we already saw the I band is the light part and the A band is the dark part. And then when we go from the middle of one I band, where that thin dark line was in the middle of the I band, to the next thin dark line in the middle of an I band, then we have a unit called a sarcomere. So the sarcomere is going to go from this zigzaggy line called a Z disc to an adjacent Z disc. So this is one sarcomere, that is one unit of shortening that we have along a myofibril. Now there's going to be thousands of sarcomeres along a myofibril. So if each one is pulling towards the center, every single sarcomere pulling towards the center then eventually is going to bring both ends of the muscle together. But remember that there's only one end of a muscle that can move, that's the insertion, and so the muscle will end up pulling the bone that the insertion is on toward the origin, and then anything else that's connected to that bone. So whatever you're lifting, whatever you're pushing, whatever, as long as it pulls on that bone, then that force will be transferred. So the basic unit of muscle contraction is a sarcomere, and we're gonna look a lot more about sarcomeres in just a minute. And then we're gonna draw one ourselves. So, so we'll do that too. All right, we're gonna go deeper into a sarcomere, and we're gonna go clear down to this level right here. And this is where you can see more things like the H zone and the M line and the thick and thin filaments. All right, so here again is one sarcomere. So I have my Z disc. Okay, so my zigzaggy line, and then I have attached to my zigzaggy line, I have a thin filament. So this blue thing made up of so 2 strands of little circles stuck together is called a thin filament. And then the red that's found in the center is the thick filament. And then we have a thin filament, alternating thin and thick and thin and thick and thin. And all thick filaments are surrounded by thin, because you've got to remember, we're just doing a cross section. I mean, a longitudinal section. If we did a cross section, which I'll show you in the next slide, you can see how there are thin filaments all the way around the thick filament. Okay, now, when a sarcomere is at rest, when a muscle is not contracting, there's a distance, there's a space between the 2 thin filaments here in the middle. We call that the H zone. We're going to go back to the picture I just showed you, because more of those things are labeled on there. And then that we have in the very, very center of a sarcomere, we have this anchoring of the thick filament proteins in what's known as the M line. So let's go back to this picture so that we can see, oh, here's H zone and then M line as well. All right, so here was our I band. And if you notice in the I band, there are only thin filaments. There are no thick filaments at all in the I band. And because they're thin filaments, that's what makes it light. If we look at the A band, it's going to extend the width of the thick filament. And the width of the thick filament never ever changes. And the same with the width of the A band, I mean the thin filament, that never changes as well. But the area where the thick and thin filaments overlap one another right here, so there's lots of protein in here, which means it's going to be dark, then this is the dark band, and it's going to extend from this thin filament or thick filament all the way through the length of the thick filament over to the other side. So there's one A band bordered by 2 I bands. And then here's the H zone in the center where the thin filaments are not touching one another. And then here is the M line in the very center of the sarcomere, in the middle of the H zone. All right, so now how does all of this work together so that we get muscle contraction? Well, let's look one more thing at if we did now a cross section, because before this was longitudinal section. So now we're going to cut through it right here, well, at each one of these little areas, and look at it end on and see what we see. So if we look just in the I-band, remember the I-band is light, and so it's only going to have thin filaments. So here are all of those thin filaments kind of forming circles around here like that. The H zone, because that's only thick filaments, we're just going to see this portion. Okay, so we'll just see thick filament and no thin filaments. The M line is what holds all of the thick filament proteins together, and so here are the linker proteins that are going to hold the thick filament together at the M line. And then if we go to the A band, you can see this is where the thick and thin filaments overlap. So here is a thick filament totally surrounded by thin filaments. Okay, and then this thick filament is going to pull on this thin from one side, and this is going to pull from the other. So we have lots and lots of pulling that's going to go on. Sometimes we refer to the thick filament as the myosin filament because of the protein that's contained within the thick filament, and then the thin filament is primarily composed of actin proteins. Those were the little blue circles that were linked together and then formed the twisted rope-like structure. All right, so now let's take a look at all of the parts of a sarcomere when a muscle is totally relaxed. So when no contraction is going on, here we can see there's the Z disc and the Z disc, so right here and right here. And then we have the I-band. So I-band's going to go out to the thick filament. So there's the I-band is light, and on this side too, because there's only thin filaments in there. But then we have the overlapping thick filament right here with the thin filament. So from here to here would be the A band, and then here is the H zone. So this area actually extends out to here. Here's the H zone, and then here's the M line in the center. Now, if you notice that these proteins, these myosin proteins, have these little heads that are going to reach out and grab onto the actin molecules and then pull them toward the center. So these heads are going to pull in this direction, and then these heads are going to pull in this direction, so that ultimately when the muscle is fully contracted, oh look, our H zone totally disappeared because the thin filaments now were brought towards the center, and the width of the I-band has gotten shorter because we moved the Z discs towards one another. So where's the space that we can lose? Well, it's in the H zone and it's in part of the I-band. Our A-band distance never changes. So the A-band width always stays the same. So the I-band will get narrower and the H zone will actually disappear in a fully contracted muscle. All right, so if we again take a look at the components of the thick and thin filaments, we can see a little bit more detail. There are myosin heads and the thin actin proteins or that form these thin filaments. And notice it says in the center of the sarcomere, the thick filaments lack myosin heads. Myosin heads are present only in areas of myosin actin overlap. So they're never going to be in this area right here, okay, when it's relaxed. And then here is a relaxed sarcomere again. And so here's the thick filament, here are the little myosin heads, and they're reaching up and grabbing onto the actin. Most of the time, because we have nervous signals that are always going to our brain, always coming from our brain, going to our muscle, that our muscles are always partially contracted all the time. We call that muscle tone. But it's not, you know, like super tight. It's just to hold everything in place. So this is a nice, I like this picture. So thick filament, thick, thin, thick, thin, thick, thin, thick. All right, now what are the components of a thick filament? Well, they are made out of a protein called myosin, and myosin is the only protein that we find, the only thing that we find within the thick filament, but we find hundreds and hundreds and hundreds of them. And one myosin molecule looks like two golf clubs where the shafts have been twisted around one another in the tail, and then they have two heads, like golf club heads, that come out. And then there are three important parts on a myosin molecule. First, we have an actin binding site, because that's the part that's going to grab onto the little blue circles and pull them when contraction happens. In order for that pulling mechanism to occur, we need ATP. So there's where ATP is going to bind. And then we have this flexible hinge region. because we need the myosin heads to be able to swivel back and forth in order to produce a contraction. So you can see these guys are in what we call the high energy configuration. So they're sticking up so they can grab onto the myosin and pull it towards the right in this particular case. Other ones have already done the pulling and they're laying back down and they're waiting, excuse me, for another ATP molecule to come in so that their heads go back up to this shape, this high energy conformation, so that they're able to grab onto actin. All right. So here are, again, here is the thin filament now. Actin is a globular protein instead of a fibrous protein. So it's just like a little blueberries with little bites taken out. And the place where the little bites taken out are perfectly designed for these myosin heads to fit in. And so when myosin reaches up and grabs onto the actin, it's going to grab onto these active sites right here. Now, we don't want muscle contraction just to happen any old time, because you know what it feels like when you got an eye twitch, when your muscle is twitching in your eye and you aren't telling it to do that. It's like, what the heck? Stop twitching. I don't want that to happen. So yeah, we do not want muscle contraction to happen when we don't want it, because we would be smacking ourselves in the face or falling down. Who knows what would happen? And so we have to prevent most of the time, until our brain tells our muscles to contract. Most of the time we have to tell them, we have to keep the muscles, the myofibrils, from connecting to one another and pulling on each other. So how do we do that? Well, we cover over the myosin binding sites on actin with a long fibrous protein called tropomyosin. Okay, so tropomyosin is going to cover over all of these actin binding sites. And then attached to the tropomyosin are some little yellow, they're not yellow in real life, but these little yellow proteins, and they are attached to they're called troponin, and they are attached to the tropomyosin. So when you pull on the troponin, then the tropomyosin pulls out of the way. And we're going to watch all of that happen and how that's going to happen and everybody that's involved in that in just a minute. What I want you to do right now is draw your own, oh, I don't know why I did that. Go back to here. Okay, is draw your own, yes. Camilla just asked, muscles always pull towards the origin. Yes, they absolutely do. They always pull towards the origin. Okay, so we're going to look at my whiteboard right now, and we are going to draw a myofibril. No, we're not. We're going to draw a sarcomere. So grab a piece of paper or write on your side of your yellow packet or down at the bottom, because there's plenty of room. We're going to draw together a myofibril. I mean a sarcomere. Okay, so let's see. Well, we'll do kind of the same colors that they were using. Okay, so the first thing, and then we'll label everything. So the first thing that we want to draw is the Z disc. So here's a Z disc over here. Boom, boom. That's probably plenty. And then we're going to do the same thing on the other side. And then if you can make your kinks line up, then that would be a great thing. Oops, they didn't there. Oh, I'm off. Dang it. No, I don't like that. And here's why I don't like that. Okay, so I'm gonna put in my my thin filaments. Okay, so here's my thin filament. So there's my thin. And then one's from this side. Oops, yeah. Okay. And then I want to put in my thick filament. Oops. And so here is my thick filament. So see how they go in between. Okay, so there's all, and you can color that in, but I'm not going to color it in. Okay, So those are all the actual physical structures. So now we're going to name things. Okay, so we're gonna go out. Whoops, I took that the wrong way. Sorry. Undo. Has to go, you point that way. Okay, so we're gonna put pointers in, and then I'm gonna put labels next to the pointers. Let's see, I think I'm gonna write those on there. Okay, so let's go ahead and label that. This is a Z-disk there, and then over here we have a Z-disk over here. Okay, and then let's see, I need another color for my text. Let's make it green. Okay, so now right here, I'm gonna put thin filament, and then I'm gonna say that it's actin. And then in here, I'm gonna put that this is a thick filament, and it's made out of myosin. Whoops, myosin. Okay, now we need to draw in our boundaries. So, oh, actually, let's do that, and let's do that in pink. All right, so from there, and then there would be one over here. Actually, I'm going to draw thin filaments out this way too for the next sarcomere, and then draw one right here. Oops. So this is too thick. This went too far. These should all line up. So I'm going to erase just, oh, like I'm going to erase it all the way, I guess. And then there we go. Okay. So you can, I should fix that one, but I'm not going to. All right. And then we would have a line over here, here, and our thin filaments going out like that. Okay, so this region that's going to be from here to here is the I band. Should have typed it out. And then from here to here is an A band. And then from here to here, I'm going to type that, is a sarcomere. All right, now what else are we missing? Well, we've got to put in, let's pull this, oops. Let's pull this out a little bit farther. Oh, dang it. Oh well, pull this out here a little bit farther here. Okay, so then we have, and I'm just gonna draw a straight line down. Okay, so from there, let's pull these to touch it. And here, in this region right here, would be the H zone. Okay, and then we got one more thing. We'll put it in dark and make it fat. Okay, so right, well, it's not very dark. Right there in the middle, then would be the M line. Okay. Oh, I see why I did that. All right, so hopefully, I know my ugly picture, but that gives you a little better idea, if you draw it yourself, the parts of a sarcomere. So you need to make sure you understand the parts of a sarcomere so that you can get now the process that we're gonna watch. So we're gonna go back to my PowerPoint, and we're gonna watch this crash course, and we're going to watch it together because we need to, so because I'm going to be stopping it along the way. So okay, for this to pop up. All right.
Romeo and Juliet, Helen and Paris, Tristan and Isolde. These famous star-crossed lovers bring to mind insatiable longing, forbidden love, and tragic separation, and poets and emo rockers love them for it. But you know where else you can find a nice hot romance? Your muscle cells. They've got their own famous coupling, a pretty pair of tiny protein strands called actin and myosin. Romeo and Juliet may have set a chain of tragic events into motion with their infatuation, but deep down in the cells of your muscles, the hot protein action between actin and myosin is actually literally causing all of your motions. All of them. And I don't just mean voluntary stuff like walking down the street or moving your mouth so you can talk or chew chips, because your muscles also support your weight and help fend off gravity. The amazing thing about your complicated, self-healing, blood-guzzling muscle tissues is that they turn chemical potential energy into mechanical energy, or movement, simply by doing two things: contracting and relaxing. And that contracting and relaxing is exactly what's fueled by the constant coupling and separation of biology's greatest lovers. Somebody get these proteins a movie contract. You will recall from our early lessons on tissues that you're kept alive and moving by three types of muscle tissue: smooth, cardiac, and skeletal. Your smooth muscle tissue is found in the walls of all your hollow visceral organs like your stomach and airways and blood vessels, where it involuntarily and very usefully pushes fluid and other material around by contracting and relaxing over and over. Your heart is so important that it gets its very own muscle tissue type, cardiac muscle, which looks striped or striated and also functions involuntarily to keep your blood pumping without you having to think about it. But when you hear the word muscle, you're probably thinking of the kind that you see on Chris Evans when he first walks out of that machine in Captain America. And those types, the ones you can see and feel and flex, are your 640 skeletal muscles. They're striated like cardiac muscle tissue, but they're also mostly voluntary, meaning you have to think about using them and activate them with your somatic nervous system. Most of them attach to your skeleton and create movement by pulling bones in different directions as they contract. Each one of your different skeletal muscles, like your biceps brachii or vastus lateralis or gluteus maximus, is technically its own organ made up mostly of muscle tissue, but also of connective tissue, blood vessels, and nerve fibers. And because your muscles are voracious energy hogs, each one is rigged up with its own personal nerve to stimulate contraction and its own artery and vein to keep it well fed with all the blood and oxygen and nutrients it needs to operate. But to understand those operations, we first need to get a grip on the anatomy of a skeletal muscle, which involves fibers within fibers and lots of layers. Basically, a skeletal muscle is constructed like a really sturdy piece of rope. Thousands of tiny parallel threads called myofibrils squish together to form muscle fibers, which are your actual muscle cells, cells with mitochondria, multiple nuclei, and a cellular membrane called a sarcolemma. Those muscle fibers then form larger string-like bundles called fascicles, which combine to form the larger rope-like muscle organ like your biceps brachii. Overall, this bundles of bundles configuration makes muscle tissue fairly sturdy, but considering how much abuse your muscles take when you do something like pretty simple, like lift a big bag of dog food, it's no surprise you need a little help. That's why every muscle contains a few different kinds of supportive sheaths and connective tissue, the protective reinforcements to keep that bulging muscle from bursting. So that is the structure part of the story. But if you want to get into the nitty-gritty, the down and dirty of how you actually move, well, there are rules. Really just two main rules, and they have to do with proteins. And they're both true for a lot of the proteins we talk about, whether they're enzymes or ion channels or receptors or muscle proteins. And these rules are: 1, proteins like to change shape when stuff binds to them, and 2, changing shapes can allow proteins to bind or unbind with other stuff. So keep those rules in mind while we see how a muscle fiber contracts and relaxes. Now, remember those tiny myofibrils that bundle up to form your muscle fibers? They're divided lengthwise into segments called sarcomeres, which contain 2 even tinier strands of protein, 2 different kinds of myofilaments called actin and myosin. And it's their angsty story of star-crossed love that fuels every movement your body could possibly dream up. A sarcomere contains both thin filaments, made up mostly of 2 light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line at either end, which is just a border formed by alternating thin filaments in a kind of zigzag pattern. A muscle contracting is all about sarcomeres contracting, bringing those Z-lines closer together. All right, so now comes the romance. When your muscle cells are at rest, your actin and myosin strands don't touch, but they really really want to. Specifically, that club-headed myosin wants to get all up close and personal with the actin. When this happens, and it will eventually, it's called the sliding filament model of muscle contraction. But in the meantime, like in any good love story, the pair have some obstacles to overcome. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way. Luckily, these guards can be bought off with a little ATP and some calcium. I prefer cash and nachos, whatever. Remember, ATP is kind of like molecular currency. It contains chemical energy, and your muscles are all about converting chemical energy to motion, so they're always hungry for more ATP. Your muscle cells have lots of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is to crank out ATP. And muscle cells also have their own version of an endoplasmic reticulum, the cell's transport and storage system, but in this case it's specialized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to save up a bunch of calcium ions. And it's also studded with calcium channels that are linked to voltage-sensitive proteins in the membrane of the muscle cell. Say I want to move my arm. My brain sends an action potential along the motor neuron until it synapses with a muscle cell in my arm. The receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which, if it's strong enough, causes nearby voltage-gated sodium channels to open. Now, I want to take a second and point out here that we're still talking about an action potential, but not in a neuron. This is happening in a muscle cell, people. So that action potential zips along a muscle cell's membrane, the sarcolemma, which has lots of tubes that run deep inside the cell called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. When those channels are thrown open, the calcium stored inside rushes into the rest of the cell, and finally myosin is like, yes, here we go! At this point, the myosin is totally stoked, because the bodyguards that have been frustrating it are in for a big, irresistible distraction. That's because the protein troponin just loves to bind with calcium. And remember, when stuff binds to proteins, the proteins change shape. So the calcium latches onto the troponin and causes it to pull the other bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. And suddenly it's all, okay. But the only myosin heads that can bind to those newly exposed sites are the ones that are ready for action. That is, the ones that have already grabbed a molecule of ATP that's been floating around and broken it down into ADP and the leftover phosphate. When a myosin head does that, it moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the energy that was released when they were broken apart. So after all that, with the myosin primed for action and the bodyguards out of the way, the myosin finally binds to actin, and it is beautiful. When they bind, the myosin releases all that stored energy, and in the excitement of it all, the myosin changes shape. It pulls on its precious actin strand, kind of like pulling a rope hand over fist. In the process, it shrinks the whole sarcomere and contracts the muscle. That's the sliding part of the sliding filament model. Now, with its energy spent, that little head has no use for the ADP and the phosphate, so they unbind with it because, remember rule number 2, changing shape encourages proteins to bind or unbind with stuff. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin in a tear-jerking scene like some microscopic recreation of the finale from Titanic. But fear not, this epic is not quite over, because this is when the myosin breaks down its new molecule of ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. And meanwhile, those calcium pumps are working hard to restock the calcium in the sarcoplasmic reticulum, so they start grabbing the calcium that's floating around, causing calcium to unbind from the troponin. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back into place. It's a circle, or potentially a big Hollywood franchise with lots and lots of sequels. It keeps repeating itself many, many times every moment while I sit here and talk, and while you sit there and eat and text and take notes, the whole drama replaying itself over and over. Kind of like you'll have to play this video over and over again to get all the little steps of the sliding filament model straightened out. But hey, some stories get better the more you hear them. If you do watch this one again,
okay, so let's go back to my whiteboard and hope that my little, yep, there's my little drawing. Okay, so if we put it into, now this is the sad part for you guys because you don't get to act this out. we actually acted it out on Thursday, and we always act it out. So we have people that are the actin molecules, we have people that are myosin, we have tropomyosin in the way, we have the Z discs, we do everything in real life, and it's fun. But I should have videoed it. One of these years I'll video it and then people can watch it, but it's much funner when you're doing it in person. But we need to go and use that same kind of scenario, because I always talk about relating it to a star-crossed lover story. And so, yeah, our myosin is Romeo, okay, and then our thin filament is Juliet, and they always want to be together. Romeo is always trying with his myosin heads to reach up and grab onto beautiful Juliet. And so she's going to be right there. But her father is very jealous, and he will not let Romeo and Juliet get together. So he is tropomyosin, or, whoops, he is Mr. Capulet. And then, so he's going to be blocking over Juliet, making sure that Romeo cannot get with her. But you know what? He has a wife that's kind of sentimental, and she loves her daughter, and she knows how much her daughter loves Romeo, and she wants to be with him. So, oh, but first, we have Mr. Capulet has a handle on his jacket or on his pants or something. And then Mrs. Capulet is calcium. And so here's what's going to happen. Capulet. Okay, so when it's time for muscle contraction to happen, then the calcium, Mrs. Capulet, will be released from the sarcoplasmic reticulum, and she'll grab onto the handle. Maybe it's a belt loop or something on Mr. Capulet, and then she's going to pull him out of the way so that Juliet and Romeo can actually be together. And then when it's time for them to be apart, then Mrs. Capulet will go back into the house, and the handle will be released, and the tropomyosin will be moved back into place, and they will not be able to bind with one another anymore. So they only get to do that when we tell them that they can do that. So that's how that works. So now we are going to go into, let's see, back to my screen share, back to my PowerPoint. Okay, so now I hope that you downloaded the little paper thing that I wanted you to download, because these are the steps of muscle contraction. And then also when Thursday was doing this, they had little strips of paper with these sentences on them, and then they had to arrange them into the right order. But we're going to do that together right now. So hopefully that you printed this out. If you didn't, make sure that you print it out some other time and then go through the steps. It would be nice if you went through the steps on your own and then compared it back, just for practice, because then that will make you understand this better. So Hank just went through all of these steps. So we're going to go through them, and I know I'm going to do it for you. It'd be better if you guys played along. I mean, if you want to play along, but you probably don't want to. So I'll just do it for you, because I know how you guys are. No, I mean, not you guys particularly, just students in general. Yeah, do it for us, Miss G. All right, so we're going to start out. What's going to be the very beginning step in muscle contraction? Well, it's going to be this one right here. Oops. It's going to be this one right here. Draw. Uh-oh. Let me redo this. Because when I don't get my thing, oh, there we go. Okay, now it'll work. Okay. Okay. Except not there. All right, stimulus begins in the brain. That's step number one. So then step number two is that the stimulus is going to travel down the nerve, because it's a skeletal muscle, and so it has to have a nerve that comes from your brain down a nerve to get to the muscle. Okay, then the nerve is going to come in contact with something called a motor unit, which we will be looking at here shortly. Motor unit is not on your list, but I'm going to have you add motor unit when we get to that in a little bit, but we are going to watch something on motor unit here shortly. All right, so then what's going to be the next thing? Well, we need to have, because the neuron does not touch the muscle cell, we need to have something that's going to cross the synapse, and that's going to be our neurotransmitter acetylcholine. So there, acetylcholine is going to be released from the vesicles, which are little bubbles. So the end of our axon looks like that, and it's going to have a bunch of little circles inside of it. And next week, when we talk more about muscle contraction, we'll talk about how's this stuff that's inside here. So the little neurotransmitter, how does that actually get released? It's through exocytosis, but we'll talk about that on next week, on Monday. Okay, so then acetylcholine is released from the synaptic vessels. It's going to cross the synapse or the neuromuscular junction and bind to the sarcolemma. So now it's going to open up the ligand-gated sodium channels, which means that the sarcolemma is now depolarized. And then following depolarization, because that's going to happen in one spot, then we're going to depolarize the next and the next and the next, and we're going to get an action potential. But first we need the action potential to spread over the sarcolemma, over the cell membrane of the cell. Then once it has traveled down the outside of the cell, then it's going to find, or it's going to end up passing down through those T tubules. And then the T tubules, or the T system, is connected to the sarcoplasmic reticulum. Remember, he said it has its own fancy little endoplasmic reticulum. So now the sarcoplasmic reticulum will get depolarized because it has voltage-gated channels in it. And then the calcium will be released. So step number 10, the calcium is released from the storage sacs. And then once the calcium is in the cytoplasm, then it's going to combine with the troponin. And then once the calcium and the troponin bind with one another, they will pull the tropomyosin away from the myosin binding sites. Now, at this point, remember Hank said that you've got to have ones that myosin heads that are primed and ready to go. So what does it take to be primed and ready to go? Well, you need ATP. So those ATP heads have to break down the ATP with an enzyme in them called ATPase. So this is the next step, is that the heads are primed and ready to reach up and grab onto the actin, and then we'll form cross bridges where the myosin heads are actually binding to the actin filaments. And then we will, the myosin cross bridges will pull the actin filaments together, sliding towards the H zone. And then in order for them to let go of the first actin molecule that they grabbed onto, then we need another ATP molecule to come in. And so new ATP molecules will bind with the cross bridges to detach from actin, and then we're going to attach to a new actin so that the process can repeat itself over and over again. Now, the only limiting factor in this case, besides how much calcium is present, is how much ATP is present. And so we want to make sure that we have a ready source of phosphate so that we can reassemble. Because remember, with ATP, when we use ATP, it becomes ADP and then phosphate by itself. Okay, so we need to build a new ATP molecule, so we have to get phosphate from somewhere. So we have a molecule in our cells called creatine phosphate. And so what that's going to do, so we'll put creatine phosphate over here. This could be, I can't remember Bromeo's friend. Anyway, I'm going to put in wrong Shakespearean characters. And so he's going to donate this phosphate right to here, and then we'll make new ATP with this creatine phosphate. So generally, under normal circumstances, we don't run out of ATP because we have plenty of stuff to make it from. It's only when we're really going through extreme events that we'll run out of ATP and we get muscle fatigue. Okay, So there is that step. Okay, so that is sliding filament model. So we've gone from sarcolemma all the way down to troponin and how things worked, the little steps and everything. So you need to review that a lot because that's part of what our quiz is going to be on. So now we're going to take a look at what is the rest of the information that the quiz is going to be on. So from twitch down to incomplete tetanus. And so you have to erase all that so I can turn the page. And so now we're going to go to the next video.
You've probably heard somebody refer to a really difficult, onerous task as the heavy lifting. Or maybe when someone else tells you that you have to do hard work on your own, they'll say, you can't have somebody else do your push-ups for you. So yeah, often when we're talking about hard work that we just don't want to do, we use metaphors that involve the skeletal muscles. And yeah, that's their reputation. They're what you use to perform all the necessary but sometimes unpleasant brute force exertions that life requires of us. But they do a lot more than just heavy lifting. Your skeletal muscles, 640 in all, come in all different shapes and sizes, from the longest, the sartorius in your upper thigh, to the biggest, the gluteus maximus in your butt, to the tiniest, the stapedius in your middle ear, which I've been doing my best to work out lately, but I just can't get any definition. These organs are capable of a whole range of power and duration, as well as surprising and delicate subtlety. The same muscles you might use to pluck an eyebrow, or catch a firefly, or cuddle a kitten, can in other circumstances crush cans, or punch holes in walls, or do a bunch of push-ups, which, by the way, are not really a thing. And she's gonna prove it. That's right, I'm gonna have somebody do my push-ups for me. When you look at how the muscular system moves, you gotta keep two things in mind. First, muscles never push, they always pull. Now, how can that be, since Claire here is obviously pushing herself up? Well, remember that most skeletal muscles extend over joints to connect to at least two different bones. That's why they're skeletal. When a muscle contracts, the bone that moves is called the muscle's insertion point, and the muscle brings the insertion closer to the bone that doesn't move, or at least moves less, and that's called the muscle's origin. And that movement is always a pull, with the insertion bone being drawn toward the origin bone. And when you think about it, it has to be this way. Muscles can't extend themselves beyond their resting state to push a bone away from it. So even though Claire's pushing herself up off the ground in an exercise we call push-ups, her muscles are actually pulling their insertions toward their origins. When she pushes herself up, her pectoralis major is contracting, pulling its insertion point, which in this case is the top of her humerus, toward the immobile origin, which is her sternum. Every single movement that your skeleton makes uses the very same principle, whether you're hammering on an anvil or lifting your pinky to sip a cup of tea. So that's the first thing. The second big thing to remember about skeletal muscles is that whatever one muscle does, another muscle can undo. You can generally classify skeletal muscles into four functional groups depending on the movement being performed. For example, the muscles that are mainly responsible for producing a certain movement are called that motion's prime movers, or agonist muscles. So when Claire does jumping jacks, she's using those pectorals in her chest and latissimus dorsi on her back to adduct her arms back down to her sides. Put another way, those are her prime mover muscles for adduction. At the same time, there are antagonist muscles that are working in reverse of that particular movement by staying relaxed or stretching or contracting just enough to keep those prime movers from overextending. So in this case, the antagonists of the jumping jacks would include the deltoids on top of her shoulders, which among other things help her slow down her arms so that she doesn't slap her thighs too hard. But when it's time to start abducting her arms from her side to over her head, those deltoids now become the primary movers, while the pecs and lats switch to being antagonists. The third functional muscle group is your synergists, and they help the prime movers usually either by lending them a little extra oomph or by stabilizing joints against dislocation. With all these arm movements, most of the rotator cuff muscles like the teres minor or the infraspinatus are acting like synergists. So this is how skeletal muscles are functionally grouped. But what about their actual functions? As individual organs, how do they contract to create both quick and sustained movements and to regulate force? How can Claire's hands gently pet this corgi in one moment and then crush a can in another? I got two words for you: motor units. A motor unit is a group of muscle fibers that all get their signals from the same single motor neuron. Since all those fibers listen to only one neuron, they act together as a unit. In a big power-generating muscle like your rectus femoris in your quad, each of 1,000 or so motor neurons may synapse with and innervate 1,000 muscle fibers. Those 1,000 fibers together form a large motor unit, and big units are typically found in muscles that perform big, not very delicate movements, like walking and squatting and drop kicking. But other muscles, like the ones that control your eyes and fingers, which exert fine motor control, may have just a handful of muscle fibers connected to a single motor neuron. Those relationships are small motor units. And when a motor unit, no matter how large or small, responds to a single action potential, those fibers quickly contract and release in what we call a twitch. And every tiny twitch has 3 distinct phases. To understand which happens when, we got to go back to the sliding filament model. Immediately after a muscle fiber is stimulated by a nerve, when calcium ions are flooding into the sarcomeres to pull away those 2 protein bodyguards of tropomyosin and troponin from the actin, that's called the latent period. The stimulus has arrived, but no force is being produced. That's when the action is just starting. Then comes a brief period of contraction when the myosin heads are binding and pulling and releasing over and over, and the muscle fibers contract. But soon the fiber slides back down into the relaxation period when the calcium gets pumped back into the sarcoplasmic reticulum, and the actin and myosin stop the binding cycle, and the muscle relaxes. Each phase consists of a lot of little steps, and while you couldn't tell by watching my brother dance, the fact is that our muscular movements are pretty smooth. That's because one muscle can produce a variation of smooth forces called graded muscle responses, and they're generally affected by both the frequency and strength with which they're stimulated. So say Claire's trying to lift something heavy, like a paint can. Just as the volume of a sound corresponds to the frequency of action potentials from your ear to your brain, her brain gets her muscles to increase their force by increasing the frequency with which her motor neurons are firing. It's like pushing a button over and over again really fast. Lift! You can do it, feel the burn, or whatever. And the faster these nerve impulses fire, the stronger each successive twitch gets, since the muscle doesn't get a chance to relax in between. Because remember, the relaxation period of a twitch is when the calcium is being pumped back into the sarcoplasmic reticulum. If another action potential travels down before that can happen, even more calcium gets released, which ends up exposing more actin for myosin to bind to, and that means more force in that fiber. In this way, twitches end up adding to each other as they get closer together in time, and that's what we call temporal summation. At some point, though, almost all of the actin binding sites are exposed, so all of the myosin heads can work through their cycles of ATP and AVP, and the muscle force can't increase anymore, even with faster action potentials and more calcium. When all those little twitches blend together until they feel like one gigantic contraction, that's called tetanus. At that point, any person on the planet will hit a ceiling of maximum tension. Now, tension means myosin and the calcium pumps are burning up the muscle cell's ATP, and the finite supply of ATP is what makes it impossible to maintain vigorous muscle activity indefinitely. Prolonged contraction leads to muscle fatigue, and when your muscles just can't take it anymore, all that tension crashes to zero. And remember, all of this twitching happens in individual motor units. Since twitches are driven by action potentials, and action potentials only have one intensity, frequency is the only way to create a grade of force. So when we zoom out to the complete muscle of maybe 1,000 motor units, we can increase the strength of the stimulus by sending action potentials to more motor units. If amping up frequency is like hitting a button again and again, then increasing the signal strength is like smashing the whole keyboard. Forehead. Since multiple action potentials don't travel down all the motor neurons at exactly the same time, each motor unit twitches at slightly different times, which helps smooth out the twitchiness. So contractions intensify as your motor neurons stimulate more and more muscle fibers. This is a process called recruitment, or multiple motor unit summation, and it's where some of your muscles' more nuanced abilities come in. So let's say Claire is holding Abby. She wants to hold on to her tight so that Abby doesn't fall, but not too tight, right? So to increase the contraction force and tighten her grip, she can recruit another motor unit. Recruiting one with 20 fibers will firm her grasp, but calling on one with 1,000 fibers might, well, let's not think about that too much. Lucky for our corgi friend, this recruitment doesn't escalate at random. It follows what's known as a size principle. It starts when the smallest motor units with the smallest fibers are activated by your most excitable neurons. Then some larger motor units with larger fibers are enlisted, increasing the strength of contraction. And finally, if you want to give it all you got, which you don't in Abby's case, your largest motor units with your biggest muscle fibers will get involved. These big guns are the last to join up, in part because they're controlled by your largest and least excitable motor motor neurons. But when they're in, they are all in, packing 50 times the force of those smaller fibers. So the basic rule is the more motor units recruited, the greater the force that's generated. Now that we know how muscle contractions happen, let's look at our two main flavors, isotonic and isometric. Say I want to pick up my crash course mug. I can do this workout myself. If the temporal and recruitment summation triggers enough muscle tension in my arm to overcome the weight of the load and lift the mug, changing the length of the muscles involved during contraction, then that is an isotonic movement. Now, if I tried to pick up a building, I could contract my muscles all I wanted and develop a lot of tension without actually changing the muscle's length, in which case I'd be experiencing isometric contractions and possibly a hernia, which is why I asked Claire to do all the heavy lifting in this episode. Today, you learned how skeletal muscles work together to create and...
Okay, so you might want to watch that a few times on your own, but I'm going to go through all of those parts and then we'll be finished. Okay, so let's go on. Now, I want you to put in, actually, let's go to my whiteboard first. Well, let's look at this, and then I want you to write down a definition for motor unit, because I didn't put motor unit on your list. It's on Thursdays, but I forgot to put it on yours. All right, so here is the concept of a motor unit. So here are two separate neurons that are coming down from, oh, and we talked about this in lecture before we left. So here are two different neurons. Here, this purple one is just going to 2 cells, but this red one is going to go to 3. So obviously, the small motor unit is going to be more easily controlled but not produce as much force as the bigger motor unit. Oh yeah, we already did this. Okay, so let's write a definition in just a sec. So then here just shows one motor neuron and then all the little branches that come off of it and going to individual muscle cells. Okay, so let's go to my whiteboard. And so we're gonna clear my whiteboard and we're gonna start from, we're gonna do text. Okay, so small, whoops, motor units. Few, so we'll just put one motor neuron, and a few muscle fibers. Okay, and then this is delicate, precise movements without a lot of force. Okay, and then large motor units. One motor neuron and many, maybe even 1,000 muscle fibers. We have gross, strong we'll put gross movements with a lot of force. And so this is the whole milk bottle. When you thought the milk bottle was empty, but no, you thought the milk bottle was full, but it was empty because your brain went ahead and picked all the large motor units at once. Okay, and then you've smashed yourself in the head with your bottle. Okay. All right. So now, so we've got that. So motor units, good. I just remember what we use the different motor units for, different kinds for. Okay. Now a muscle twitch. This is one contraction after a stimulus. So we just stimulate it once. Here's my single stimulus. And then we get this period called the latent period. So make sure you write down these 3 periods with muscle twitch. So the latent period is the first one. And so what's going on in a latent period is there's no contraction. So there's no force production. So from here to here, it's like, what the heck? I've already stimulated the muscle right there at time 0. Why did it take, 5 milliseconds before anything started to happen? Okay, so this is why. So at step 6 on our list, okay, step 6 on our list says the sarcolemma is depolarized. That was this point right here. So the sarcolemma received the neurotransmitter signal from the neuron, and now became depolarized. But you can't just go right from the cell membrane being depolarized to muscle contraction, to actin and myosin binding. So if we go from sarcolemma being depolarized to cross bridges to cross bridges pulling the actin filaments together, you guys, that's steps 6 through 15. So all the calcium has to be released. Well, the signal has to travel down the sarcolemma. It has to go into the T-tubules, has to depolarize the sarcoplasmic reticulum. The calcium has to come out, bind with the troponin, pull the tropomyosin out of the way, and then the actin and myosin can bind. So we got 10 steps that have to occur in order for actin and myosin to be able to bind. And so that's what's going on in the period of contraction is, so contraction, this is latent. So the latent period is steps 6 through 15. Okay, contraction, the period of contraction is sliding filaments, where the filaments are actually sliding. Okay, and then the period of relaxation is returning back to normal. And the important thing we want to remember there is that calcium goes back into the SR and back into the sarcoplasmic reticulum, and so it's pumped back in there. So what if it doesn't? What if calcium doesn't get pumped back in? Well, then your muscle stays contracted, and that's sometimes why we get a cramp after we do really strenuous activity, is because we ran out of ATP, but yet the calcium was still present. We couldn't pump the calcium back into the sarcoplasmic reticulum, and so we got a cramp. We got muscle contraction and we couldn't let go yet. Okay. So those are the 3 steps of muscle twitch: the latent period, period of contraction, period of relaxation. So the latent period, no force production because we're going through steps 6 through 15. But force production will happen when we have sliding filaments. And so that's what's going on to here. And then now we have calcium being pumped back into the SR, and then the tropomyosin goes back into place, and then actin and myosin can't bind with one another anymore. All right. Okay, now what else did he talk about? All right, so here's what's going to happen when you get a single stimulus. The muscle contracts and then it relaxes, and that's the maximal tension for that strength of a stimulus. So we're just going to boom, contract, and then we're going to relax. But what if we continue to stimulate the muscle so that we can get greater tension being produced? Okay, that's when we increase the frequency. So we stimulate it faster and faster and faster. Okay, so even though it's low frequency, we still stimulated the muscle one after the other. We call that wave summation, or he called it temporal summation. Either one is fine. But what are we doing? Ooh, look at the muscle started to relax, but then it contracted again. There's still more calcium present. So more myofibrils and more muscle cells can contract. And then, oh, we stimulate again and more muscle fibers can contract until we get a whole lot of tension being produced right here, more than with that initial stimulus. So we stimulated it more quickly, even though we didn't stimulate it super fast, which I'll show you in a minute. But we stimulated it rapidly, one after the other, and then we get more tension resulting. So if another stimulus is applied before the muscle relaxes completely, then more tension results. This is wave summation, so that's stimulating it faster, and then we get this unfused, or what's known as incomplete tetanus. So we get this rising up to more maximal contraction here. Okay, so that's wave summation. And let's make sure we know with wave summation that okay, so we increase stimulus frequency. We stimulate it faster and faster and faster. Okay, so increase stimulus frequency. Now, what if I stimulate it super fast? Okay, then I'm going to get this next one that he showed. I'm going to get complete tetanus. So here at higher stimulus frequencies, there's no relaxation at all between stimuli. This is fused or complete tetanus. So we get lots of contraction, lots of force, and then we get no relaxation in between. Look how fast we're stimulating. Right down here, just one right after the other. Now, the interesting thing with this is that we cannot get ourselves to complete tetanus. We can get to incomplete tetanus. You know, you're holding a heavy weight above your head. You don't want to drop that on your head. You're just going to keep contracting, 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 fast, fast, fast to hold that heavy weight up there. And you know, your muscles start to quiver because they're trying to relax, but your brain's telling you, don't you dare drop that weight. And so that's incomplete tetanus. This, though, my friends, will never happen with your brain, because your brain's like, if I contracted all my muscle fibers at the same time, I could rip my muscle off of my bone. I could tear my tendon. And we always want to save a little bit back. I'll try to figure out how to show you a video. Maybe I can run it in my computer and show it to you. Of the closest that a person's really ever gotten to complete tetanus. But this is what's going to happen to you when you get tased. So if you've ever seen somebody, and that's usually with a lower frequency, but if you zap somebody with a higher intensity taser, they're going to just curl up in a, well, they're not going to curl up in a ball. They're going to stretch out, and I'll show you when we go over tetanus in lecture, what that would look like if somebody got tased. It's also going to look like what happened if somebody got the disease tetanus, because what's happening when you get the disease tetanus is that there's a toxin that's binding instead of acetylcholine. This would be like acetylcholine being released fast, 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 one little bit after another. Well, if you get the tetanus, you know, step on a rusty nail and you get or a nail, and you get the tetanus toxin in you, the toxin just stays in the neuromuscular junction. So it's as if the muscle is being constantly stimulated, so you'll go into tetany. And your diaphragm will seize up on you and you'll suffocate to death. You'll take in one last breath and you won't relax it, and then you die of respiratory arrest when you have tetanus. So make sure you have a tetanus shot. All right, so that's complete tetanus. This is incomplete. You get a little bit of relaxation in between stimuli, but you don't get any relaxation in between stimuli with complete tetanus. All right, and the last thing that we need to take a look at is multiple motor unit summation. So what is that? That's when, depending on the strength of the force, if it gets heavier and heavier and heavier, then we're going to use more and more motor units in order to hold up the whatever we're holding. So let's say that girl, Claire, was holding the dog in her arms, and she had to hold her arms out. And then they started putting more dogs on her arms. Okay, so here's just putting her arms out, you know, at 90-degree angle, just holding her arms out in front of her, her elbows bent. That would be right here. In order to hold her elbow or her lower arm, her forearm in place, it probably only takes 2 motor units. Here's all her motor units that she has to pick from. A little bit of stimulus. Yep. Okay, so then they put 1 dog on top of her. Okay, and so, ooh, that's gonna take some more muscle fibers, because, you know, maybe it's a little puppy or piece of paper, something very small, doesn't weigh very much. And then they added another weight onto her arms. Now she's going to keep using those same motor units that she already contracted, that she's already firing, but then she's going to add to them summation, multiple motor unit summation. And then we make it so the stimulus was even higher, so we added another dog. So now she's holding 3 dogs in her arms, and so now she's almost all of her muscle fibers, and let's add in another dog. And so now she's at maximal contraction. All of her motor units are firing, so she's at tetanus as well. This isn't a good showing it going down like that, but her muscles would stay sustained like that, and you can't produce more force. It's just going to stay right up there. In fact, let me draw that. Because if you got her to, and again, it's not going to come up to here. So this would come up here like this, and then it would be sustained. Even though we're stimulating her, putting more and more dogs on her, this is all the force that she can produce right here. And then finally, she's going to run out of energy and she's going to fatigue. But notice that at the maximal stimulus right here, all the motor units are firing. I Same with each one of these because I don't have any more. Okay, so how do we get multiple motor unit summation? We increase stimulus strength. So we make it harder, heavier and heavier and heavier. Okay, so I have that same. Oh wait a minute, I have to clear. I have that same graphic that he had as far as what kind of motor units do we use. So we're going to use the smallest ones first. Those are the easiest ones. And usually we don't need a lot of force. So we're going to use them first. But then if we need more force, then we'll recruit the ones with more muscle fibers attached to them. And then, and even ones that might be bigger around, muscle fibers that might be bigger around because they have more myofibrils in them. And then we'll use the biggest ones last. So we're always going to use the little ones, conserve our energy, and then call in the big guns if we need to. Okay, that was almost the last one. The very last one is a concept known as trepa. So what happens in trepa? Well, this is what we call muscle warm-up. Well, let me type that in there so that you can see that. Okay. All right, so let's say that we want to shoot a basketball, okay? And we come up to the free throw line and we shoot a basketball. Dang it, we shot a brick, okay? It didn't even bounce off the front of the rim, okay? Because my muscles weren't contracting at the most efficient, the greatest efficiency for that force. So I'm standing at the free throw line. I shoot, not so good. I shoot again, I get a little bit better. I'm not shooting any harder. I'm shooting with the same force that I know I need to make it in at the free throw line. But then I shoot a little bit more, shoot it again, I shoot again. And finally, hopefully it won't take as many times to shoot the basketball to make it in the basket. But now I shoot perfectly well, okay? Because my muscles are working at their peak efficiency as far as their enzymes are concerned. And so this is the force they should have produced way down here. But because my muscles were cold, they did not contract as efficiently as they do up here. But notice what happens between every stimulus. We contract, we go right back down to zero again. We contract, we go back down to zero. We contract, we go back down to complete relaxation. That is different than if we look at, didn't want that. Then if we look at incomplete tetanus, you can see the muscle did not totally relax in between. So that's the difference between incomplete tetanus and trepa, is that the muscle has an opportunity to completely relax before you contract it again. Okay, so that is that. And there we go. So that is everything that I have for this lab. And so hopefully that brought you up to speed, and that you should be right up there with the Thursday lab. So now, and I'll let you know when we're going to meet again for lab, which will be the next week just on the lower extremity muscles. So that one won't be very hard at all. And then we're only going to we'll meet together as a whole lab. So both Monday and Thursday people will meet together. And then, remember on Monday, we'll meet at 11 o'clock in just to start with our muscle physiology, just like we were back in the classroom. All right, so does anybody have any questions before we bug out of here? You can send it to me in the chat or not. Okay, well, if that's it, then that's it. Thanks for, oh, yeah, I will open up your assignment so that you can drop them all in the PhysioX. I mean, in the assignment Dropbox. I just haven't gotten to that yet. So that'll be tomorrow. That's my come to work and open up everything so everything can be submitted online, and all your quizzes will be online and everything will be online. So you'll be able to do that. Okay, well, if that's it, then we'll stop this.